as a recap of our discussion, so biomolecules, when we say biomolecules, these are lipids, for example, these are fats or glucose or etc. So there is what we call an arrangement hierarchy from higher to lower, from lower I mean to higher. So we have glucose, for example, to glycogen, and amino acids to proteins. When you say glucose, it is a monosaccharides. It is composed of one sugar only. And amino acids, so we will discuss later on what are the different amino acids, what are the smallest amino acids. So when combinations of two amino acids, it will form a peptide. Peptide plus peptide, it will form a protein or a polypeptide. Okay? So, as in overview of the study of metabolic process, carbohydrates, fats, and proteins are broken down during the digestion process. That is primary metabolic process or metabolism. So small units, carbohydrates are digested to monosaccharides, for example, carbohydrates to glucose, and proteins to amino acids. So there is what we call a digestion process happens. After the digestion process, so absorption follows, small molecules are further broken down and oxidized to carbon dioxide. So the nicotinamide adenine dinucleotide plus hydrogen, okay, and flavin adenine dinucleotide plus hydrogen is generated. So when carbon dioxide is produced, that is already secondary or intermediary metabolism. When you say intermediary metabolism, that is not the final metabolic process. It needs one or more metabolic process to become the final or the end process. That is intermediary. Again, NADH and FADH stands for, NADH is nicotinamide adenine dinucleotide plus hydrogen, while FADH is flavin adenine dinucleotide plus hydrogen. So secondary metabolism, so the product there is reduced, enters the electron transport chain to oxidize to water, and ATP is trapped. So there is a generation of energy in the form of ATP in tertiary metabolic process. So tertiary metabolic process might be the last part. Okay? Stabilizing forces in molecules. Covalent bond, ionic, hydrogen, hydrophobic, and Van der Waals forces. So these are, I mentioned this one in our organic chemistry. It's just a review. Okay? Ang tawag Van der Waals forces is London forces. Okay? When you say covalent bonding, there is what we call a sharing between two elements or two molecules. Example, this is the molecule of water. So there is a sharing of two electrons of water of oxygen is being shared to hydrogen. So during the chemical reactions, so hydrogen to hydrogen plus oxygen, it will form a water. Okay. For ionic, there is a transfer of an electrons. So this is between the nucleophiles and the electrophiles. When you say nucleophiles, the electron rich, electrophiles, the electron poor. So transfer of electrons, for example, sodium will transfer electrons to chlorine because chlorine has the highest number of electrons compared with sodium. Okay? Though that is ionic bonding. Next, so in proteins, chemistry charges are produced by amino group of lysine, guanidinium group of arginine, and imidazolium group of histidine. So when you say guanidinium group and imidazolium group, these are also known as the chain group that is present in the structure of histidine. Okay, so these are the positive group. So enzyme plus substrate, it will form enzyme plus substrate complex. So during that reactions, there is a positive and negative charge. So in protein, there is also a negative and positive charge. Usually the positive charge there is the amino group in proteins, while the negative charge are the carboxylic group or the COOH. Okay? Next, hydrogen bonding. So when H bond formed by sharing of hydrogen between two electron donors. When you say donors, they give up electrons. Okay? So biological system, both donors and acceptors are usually nitrogen and oxygen to form NH2 and OH. Why? Because nitrogen and oxygen are abundant in nature and they have higher electrons. Okay, they can able to accept or they can able to donate. So for this one, water, then we have the ammonia. So hydrogen can able to bond with your nitrogen. So there is a spare electrons here. Okay? Next, in proteins, the release group or donors, take note imidazole, indole, and peptide. OH, serine, and threonine, and NH2, arginine, and lysine. So later on, we'll discuss the structure of the donor group. Okay? Acceptors, because they accept proteins, usually naka-negative charge yan. Aspartic and glutamic acid. So we'll discuss the structure later on. Okay? C equals to oxygen or SS disulfide bond. DNA that is bonded to hydrogen, maintained by purine and pyrimidine. So later on, we'll discuss in DNA, there is positive and negative. Usually the H bond, hydrogen usually bonded with it certain DNA. Okay? Hydrophobic interactions, meaning the interactions between non-polar hydrophobic chains by eliminating water. So usually when they bond together, this is known to be mga non-polar or the lipid-soluble compounds because when they bond to each other, they get rid of water. So take note of this one. Okay? That is hydrophobic bonding.